And hello again. We're back to talk more about bacteria, and in this case, disease-causing microbes, microorganisms. And we have talked at length about what we know about pathogens that have the ability to cause overt disease. Those are referred to as frontal pathogens, and we know all of their tricks about how they play the game. And what I would like to focus on here is the idea that not all pathogens are created equal, that pathogenicity is complicated, and just realize that in the age of during the golden age, we learned quite a bit about especially bacteria that caused diseases, and we did such a good job during that period of time that we sort of overlooked a lot of things, especially once antibiotics were discovered and implemented widely for infectious diseases. A good deal of research stopped. And the idea that not all pathogens are the overt ones, yes, granted, they cause profound diseases, but a lot of conditions and chronic illnesses can be attributed to infectious diseases as well, by microorganisms that employ tactics that I'm going to just refer to as stealth pathogenicity. So that's what this lecture is going to be looking about. So just kind of starting with going back to that golden age thinking, right? So when it was discovered that there were microorganisms behind disease, it wasn't just breathing bad air. It wasn't, God hated you and wanted to strike you dead because you did something wrong. Once it was realized that that diseases could be caused by microorganisms, it became kind of ingrained in conventional thinking that microorganisms in general were germs, they were disease causers, we needed to do everything possible to avoid them, follow germ-free practices, wash our hands, sterilize our food, sterilize everything, live in a bubble, and that would make us all healthier. So we did that for a lot of years, especially once antibiotics were discovered and implemented, and with the presumption that getting rid of microorganisms, getting rid of bacteria, getting rid of all these different things that were found in the environment that we could be exposed to would make humans healthier overall. So again, going back to golden age thinking, and it was, this was what Koch and his fellows did, was they proved that there is one microorganism that causes one disease. You could track it using the Koch's postulates to determine what microbes cause what disease. You could prevent it by preventing transmission. So sterile practices were implemented. That took a little bit of time, but and shout out to nurses because they were often the ones that were doing the implementing. Realizing that if these were physical agents, if you prevented them from getting to another person, then you could prevent the disease. So if you could prevent transmission, then by that one microbe, then humans in general would also be healthier. So the other part that developed out of the golden age was the understanding of how the immune system responds to infections by pathogenic organisms. And, we developed a model of how the immune system is supposed to respond, starting with the innate responses. Phagocytic cells come and eat the things, they process them, and then they present them to cells in the lymph node, which then can respond in a very specific fashion. I mentioned those as, the first responders versus the Delta Force kind of approach, where the adaptive response was more Delta Force. Go in there, strategically remove the offending microorganism. And then, of course, once that occurred, humans would return to good health and then would develop long-term immunity. So secondary, third exposures to that organism would not result in life-threatening illnesses. And then came the idea that, well, we can prevent it. If this is how the immune system works, then we can prevent infectious diseases, all of them, by just triggering our immune systems, pre-triggering them to be ready for the first time that we encounter those microbes. So we don't have to go through the whole sickness thing and, do it the hard way, go through that whole illness so that we can develop the immune response and have protection for later on down the line. So all of those conventional approaches or ideas are still in practice in medicine today have not evolved much. And I think that our current situation with COVID is a perfect example of that, because the first thing that everybody decided that we needed to mitigate COVID was a vaccine. So, none of the other practices were considered worthy. And when I'm talking about other practices, I'm talking about therapeutics that were longstanding, tested, safe, effective, and yet disregarded as therapeutics because why? They were old, they were cheap, they were, waiting for the vaccine. And that's all I'll say on COVID. So with that in mind, I just want to review a couple things with regard to infectious diseases and morbidity and mortality. And I'm going to use some data here to show you this. So notice this data came from 2016. This is the World Health Organization, just so you know where this data comes from, which of course that's their mission, is to track infectious diseases. And just looking across the board at the top 10 causes of death, and they actually did this across a spectrum of countries. So low income, middle income, low middle, high middle, and high income countries. So low income countries where lack of resources, lots of infectious diseases, lots of pathogenic microbes, some that we don't even see here in the United States. The United States would fall into the category of a high income country. So just comparing those two, right? So top 10 causes of death in low income countries in 2016. Notice that we're using the mortality rate expressed as number of deaths per 100,000 people in the population. And notice across the board, these are the numbers, and then these are the diseases that were leading causes of mortality in low income countries. Notice the red are infectious communicable diseases, okay? So what were the major causes of death? Was lower respiratory infections, that's pneumonia, foodborne illness, HIV/AIDS, malaria, tuberculosis. So between all of those, that's something like 3/4 of all deaths due to disease in low-income countries are due to infectious diseases, okay? So overall, and I just look at this number here, so 440 deaths per 100,000 people in low-income countries, with 3/4 of them attributable to communicable diseases, assuming that if they had better access to healthcare, if they had antibiotics, if they had more vaccines, if they had better sanitation, if they had even better living conditions, that many of these deaths could be prevented, their lives would be better. We want to make them just like us. So let's go look at us. So here's us, all right? Going back one step, notice the numbers across the top. Okay, so we're up to 80 people per 100,000. And I just want to point out, notice the scale across the top for countries like the United States, for high income countries. So one thing to point out is that the death rate overall in high income countries is higher than that of low income countries. Okay, so more people per capita or per 100,000 die than from things other than infectious diseases than those that die of infectious diseases. Okay, so thing one to look at. Looking down the list of what actually causes deaths in a high-income country like ours. So what do you have? Cardiovascular disease. If you add all these different cancers together, that would be number 2. So cardiovascular disease, cancer. Looking on down the list of other diseases, COPD, which is lung changes that is often related to smoking or other behaviors. Lower respiratory infections, so pneumonias, is the only infectious disease, communicable disease, that makes the top 10 list. So notice overall the number is 40 per 100,000. So in low-income countries, infectious disease are 3/4 the cause of all deaths, but they're a lower rate of death. In high-income countries, more people have chronic illnesses than they die from chronic illnesses than they do from a lower, from infectious diseases. So that in and of itself says, all right, so, here we're doing a better job preventing and curing and all the rest of with infectious diseases, but look at the rate of chronic illness. And I also want to point out that many of these conditions, cardiovascular conditions, Alzheimer's disease, other dementias, cancers, it's a growing body of evidence that indicates it is a disruption in your microbiota, right? So it's a microbiome issue, dysbiosis occurs that contributes to those chronic illnesses. And in addition, that many of these diseases, now that we have the tools available to look beyond the superficial, beyond culturing, beyond, even other ways that we have of looking for causes of infectious disease, we can go looking for their DNA, their genes. It's clear that many of these diseases may have behind the scenes a stealth bacteria or protozoa or other agent that could be causing those kinds of illnesses. Many of them are chronic inflammatory conditions. What is inflammation? The immune system responding to something. And so the question is, what is that something? What is the immune system going after that's causing that chronic inflammation that's leading to all these chronic diseases? And what I'm going to tell you is that a hypothesis that is gaining more and more support is that it is stealth pathogens that are behind this ultimately. And what we're doing to contribute is completely disrupting our microbiome with our urge to eliminate all other bacteria using antibiotics, being as sterile as possible, vaccinating, and a lot of other things that go along with living in a high-income country like ours. So this may be controversial, but I'm telling you anyway, because this is a way to look at that data. And as a microbiologist, this is a reasonable hypothesis that should be explored further. So this, just looking at how infectious disease contributes to this issue of chronic illness, realize that, infectious diseases profoundly impact the human microbiota. How? Because they, what is the treatment for infectious disease? Antibiotics. They're not only killing the pathogen, the direct pathogen, they're also killing your microbiota, the ones that are susceptible to those to the different antibiotics. So infections lead to the use of antibiotics. Some of these new infectious, new emerging diseases are immunosuppressive, which also, that changes the balance of the immune system and what the microbiome is doing. Use of immunosuppressive drugs for all of these autoimmune diseases, interestingly enough, right? So you suppress the immune response, but what if those diseases are actually, they start with an infectious agent? What if they start with a microorganism, either part of the microbiome or something you've acquired? Something to consider. So now you're suppressing the immune system further. Some of them suppress the immune system on their own. So your immune system has no chance to make a difference here. Other modern treatment technologies, right? All of these things that are in response to these new emerging infectious diseases, HIV, other infectious diseases like hepatitis B, which is a chronic viral infection, that sort of thing. The treatments to that are leading to microbial dysbiosis, right? So your microbiome is becoming unbalanced, and I'm going to show you some examples of that. So what does that do? What does your microbiome do? It influences how your immune system functions. Through your vagus nerve, your gut microbiota tells your brain what's going on and how to respond to things. And that neuroendocrine, the gut-brain axis was something that I was talking about previously. Resistance to antibiotics is a growing issue. Many stealth pathogens are tolerant of antibiotics, meaning they could care less if you treat them with antibiotics. So there is a need for new therapeutics to address these types of infections. Clostridium difficile is an antibiotic-associated infection of the gut, which is the bacteria already there. What we do by killing off all their competition is allow the endospores of Clostridium difficile to germinate, leading to active infection. If you left them alone and the microbiome was doing its job, this would never become a problem. So all of these things, contribute to the chronic illnesses that are 70% of all diseases in this country, according to our own public health agencies. All right, so I'm going to propose, and I'm not the only one proposing this, in the, even infectious disease, right? Infectious disease in the age of the microbiome, so you get infected with a microorganism, but you also need to consider the other impacts of that infection, inflammation, which is part of the innate immune response, and immune dysregulation, because many stealth pathogens completely disrupt, they take off the rails our regular immune response, because that's what they do. This is how they survive inside of their warm-blooded host animals, is to interact with their immune systems and say, let's come to some sort of agreement. But that takes a long time. And as new hosts to an old pathogen, our immune system hasn't gone through that with them yet. So we are in this period where now our immune system is starting that negotiation with the pathogens. But in the meantime, they know more about us than we know about them. And so problems arise as a result of that. So, and just always keep in mind the central role of that your microbiota plays a central role in keeping you healthy. Okay, so let's look at that microbiota, because it's not just microbiota, but there's yeast, there's viruses involved in this as well. So here's the microbiota here's a typical average healthy person, and you can see these pie charts show different parts of the human body and the different groups of bacteria that you actually find there. Okay, so these are, if you remember, the Firmicutes are the gram-positive bacteria. Actinobacteria are the bacteria that have that waxy cell wall, right? The acid-fast types and others that have, they stain gram-positive, but they have other properties. Bacteroidetes, which are gram-negative, anaerobic bacteria. These guys, I want to try to say that out loud, fusobacteria, proteobacteria, these are the gram-negative, facultatively anaerobic or anaerobic bacteria that we talked about. Just to give you some examples, this here would be Staphylococcus aureus, Bacillus cereus that we looked at in lab. Actinobacteria, Micrococcus is one of those. The mycobacteria are in this particular group. E. coli is, of course, the classic example of a proteobacteria. So you can see by the colors what parts of the human body have what. So, just kind of looking at, the gastrointestinal tract. Okay, so notice, in a healthy human, the GI tract has a very high proportion of bacteroidetes, these anaerobic gram-negative bacteria that are present, followed by Firmicutes, the gram-positive bacteria, followed by proteobacteria, the E. colis of the world, okay? So, but notice what happens when so this is the normal state of somebody who's in good health. Look at, we have controls here versus patients, okay, for inflammatory bowel disease, type 2 diabetes, necrotizing enterocolitis. So, healthy control, we see a lot of those bacteroidetes, we see Firmicutes, but look at somebody with inflammatory bowel diseases. Look what's happened. Proteobacteria take over. So this is the E. colis of the world, all of a sudden come stampeding back into the picture. Not necessarily always E. coli, I'm just choosing that as the representative proteobacteria. Firmicutes are kind of getting crowded out as a result of that. But the big change, here we have tons of those bacteroidetes, and here we have a very tiny fraction of them. And we have an increase in the actinobacteria. Type 2 diabetes, the Firmicutes are decreased. The bacteroidetes are increased. So two different disease states, the same bacteria is decreased in one disease, but is increased in the other. So just to give you an idea how complicated this is. Now, type 2 diabetes is not, the same as type 1. Type 2 diabetes is more metabolic than anything. So this probably originated as a result of dysbiosis and is perpetuated as a result of dysbiosis. And then necrotizing enterocolitis, notice, right? So here's our proteobacteria, look at what happens, right? So the proteobacteria are, you know, like I said, the E. colis of the world come storming out of their hiding where they've been kept under control by these other types of bacteria, causing disease, right? Necrotizing enterocolitis, meaning the inside of your intestines is sort of being shredded. And there are bacteria, by the way, that do that. So bacterial infections can actually cause that, like Shigella infection can actually cause that, which is a type of proteobacteria. All right, so there's one example. Here's another one. This is fibromyalgia. So just kind of looking at control versus fibromyalgia. The changes in the populations of bacteria. Again, look at the list, right? Actinobacteria, Bacteroidetes, Firmicutes, Proteobacteria. And you can see where for a patient with fibromyalgia, right? This is the healthy control. Notice there are far more species, and this actually shows how much each has changed from control, right, to now all of these purple guys start showing up, right? So the controls versus the purple guys. So these are increased, and some of them are increased significantly. So in a fibromyalgia patient, by the way, what fibromyalgia was is a disease of widespread pain, widespread pain throughout the body. A lot of it is muscular, not just like, ow, I pulled something, but that feeling all the time across your whole body. So on severe days, people can't even get out of bed with this particular disease. So again, disruption, this is the gut microbiota causing or being related to, because this isn't cause and effect, there is no cause and effect here, but it is related to this disease state. And I'm going to add that in the condition known as post-treatment Lyme disease syndrome, which is also known as chronic Lyme disease, that this study published just a couple of months ago shows that there is a change in the microbiome between patients who are healthy and those who have chronic Lyme disease. So another situation where that is the case. Okay, and in this case, there was an increase in this particular species called Blaudia. Maybe you saw that on the list on the previous slides, and a decrease in the Bacteroidetes. So what the point of this is, we now have a microbiome signature that actually proves that this is a disease entity, which, believe it or not, there are still healthcare providers, medical doctors who are saying this is not a medical condition, this is a psychiatric condition where people just want to be sick, so they're sick. But now we have a distinct profile showing that there are alterations in this disease, just like there are for patients with MS, just like there are for patients with Parkinson's disease, just like there are for patients with fibromyalgia. So these widespread, systemic, multiple different types of symptoms that go along with that, there's something biological going on. It's not just somebody wanting to just be sick for the sake of being sick. Okay, so let's look at the fungi. Okay, so that's the Mycobiota. These are all different types of fungal species. Some are yeast, some are molds. And you notice that just like we have a bunch of bacteria that make up our microbiome, we have mycobiome as well. So we have yeast and we have molds helping us out also. So notice this is on the healthy side. Again, in all these different locations of the body, you can go compare these to these. But notice that in a disease states, you have changes in the microbiota as well, right? So healthy lung, you see a lot of one species. Over here, you don't see any of those. So you can look down the list and just say, whoa, so it isn't just about our microbiota, the mycobacteria, so the yeast that inhabit us, and also these mold species do too have an effect on our health, our overall health, and sometimes they cause disease. Okay, so going back and looking at that, the big takeaway message here is, so pathogens cause disease but it's really complicated. It's not just, here comes the microbe, it gets in you, it grows to large numbers, it triggers quorum sensing. Everybody in the population of bacteria turns on the same virulence genes. The toxins that they produce make you vomit, make you sneeze and cough, make you have diarrhea, so that their goal is to spread to new hosts and just keep going along those ways. Those are referred to as primary pathogens. I affectionately call them the asshats because they're just, they're just the bad guys. They're just trying to make a living, but it's at the expense of us, who they manipulate into, by the way, they manipulate our bodies into doing diarrhea. They manipulate our bodies into, like I said, coughing and sneezing, so that they can spread. That's their goal. So what are the characteristics of a primary pathogen? They have fast generation times, right? So they'll double in number in 30 minutes or 40 minutes. So in other words, their population goes from very small to very large in a short window of time. Then remember, after that, once they reach the quorum, they initiate, they send out their signals, the quorum sensing, they send out their signaling molecules to turn on. So everybody in the population turns on their toxin genes all at the same time, so that there's a profound effect on the host, which furthers their need. So these are the genes. Remember, we talked about the different types of toxins that bacteria can produce. We talked about other virulence factors, population expression of those genes. So once you hit quorum, then you attack, but don't do it before that because you might be defeated. So it's a great way, obviously, looking back at history and even to the present, of making a living. These are the guys that triggered that relatively predictable immune response that, we would like all pathogens to do. But as we study the immune system more, we realize that doesn't happen all the time. But these are the ones, so you have innate immunity followed by adaptive immunity followed by memory. Long-lived antibodies, you have protection from future infections. So those are the primary pathogens, but realize that not all pathogens do this. And even some of the ones that do it also have stealth capabilities in the background as well. So that gives us, and actually this slide I just added because I wanted to show you a reminder of the primary pathogens, all of their toolbox, if you will, their weaponry, all of the different types of things. Not all primary pathogens have all of these, but they will have some combination of these virulence factors that they will produce en masse so that they get the overall host response. And remember that their existence is predicated on their ability to spread to new hosts. Okay, so those are all the characteristics that make a primary pathogen an asshat. Then there's various shades of pathogenicity. So I kind of, think of this as, it's not an all-out thing, all-out attack all the time. There's various parts of it. So there are some microbes who are not asshats. They're just sort of members of our microbiota, but they possess the genes for those virulence factors. They just don't always express them. So those are referred to as opportunistic pathogens. So these are ones that you might acquire from the environment, or they might be ones that that are part of your own microbiota that become empowered to step up and do something as a result of microbial dysbiosis in your microbiome. So Clostridium difficile is a perfect example of that. So that's present in probably everybody's gut. Most people don't get C. diff or get necrotizing enterocolitis as a result of it. Some people do. Why? Because of dysbiosis. So I just gave you a couple of examples here, right? Staph aureus, Staphylococcus aureus is an asshat, but on the other hand, it needs to get into the body before it, expresses those superpowers. When it's on your skin, it's more or less a benign microbe. When it gets through the skin, then it can start, becoming more mean, if you want to look at it that way. Haemophilus influenza, Streptococcus pneumoniae. Okay, these are members of our respiratory microbiota. And yet these two are major causes of pneumonia. And by the way, in children, these are major causes of meningitis. So they actually have the ability to cross the meninges and get into the brain and cause meningitis. And then pathobionts, this is a relatively new term used to describe our members of the microbiota that can cause disease in population level ways, right, that are mostly associated with chronic inflammatory conditions. And here's a listing of these. The only ones that you might recognize here, right, Proteus mirabilis and Klebsiella pneumoniae, these are you know, those gram-negative enteric bacteria that we looked at in our lab about that. Clostridium difficile, as I mentioned previously, Enterococcus, these segmented filamentous bacteria that are throughout our gut, which, you know, many thought had a protective role, but it goes back and forth depending on other conditions. Helicobacter pylori, which is an inhabitant of our gastric mucosa, is a microbiota that can cause chronic inflammation leading to ulcers, and stomach cancers, in fact, can go along with that. So primary pathogens are the asshats. Opportunistics are the, think about it, there's just like, oh, look over there, I can get over there and cause disease, so I'm gonna. That's an opportunistic pathogen. And these guys are just the ones that are living there, and then conditions change, so that results in a response which can lead to chronic illnesses. Okay, so obviously it's complicated. So there are at least 50 shades of pathogenicity. And where I want to go from here is to talk about those that are mostly in the stealth range, the ones that are not overt, they're not you know, out there to spread. They have completely different goals in mind. So to achieve those goals, they have to be able to somehow manipulate our body's response to their presence to promote their own long-term survival. So I've listed some of them here. We're looking at some of these in lab, just to kind of give you a list. Syphilis is one of those diseases. Chlamydia this is a bacterium, this is a spirochete bacterium, that even though, you know, syphilis has been around for a pretty long time, but nonetheless, we still have it, and now it's becoming drug resistant. Chlamydia is the #1 reported infectious disease in the United States at the moment. This is sexually transmitted as well, causes persistent infections. Many people are infected with chlamydia and have no knowledge of it, which is why it spreads so readily, because asymptomatic people having sex with other people spread it that way. The rickettsias and the anaplasma. Anaplasma we looked at in lab most recently. These are the ones that invade and reproduce inside phagocytic cells. There's also a hypothesis that these guys might actually be the ancestors of the mitochondria, because remember, that was an endosymbiotic type of bacteria back in the day. And when you look at their DNA, they trace back to this particular group of proteobacteria called the alpha proteobacteria. Tuberculosis, right? Mycobacterium tuberculosis, Borrelia burgdorferi, and other Borrelia species, this is the cause of Lyme disease, and other Borrelia species cause diseases called relapsing fevers. So all of these are examples. I'm not going to talk about all of them, some of them, but not all, but these are different, okay? Many of them are drug resistant. And actually, I should say they're drug tolerant. They live in dormant states where antibiotics don't even touch them. And so they're outside of our rules. The rules of infectious disease medicine, these fall well outside the range of it. And as a result, there are no effective treatments. They're very difficult to diagnose, and nobody really knows what to do about the chronic illnesses that can be associated with infections with these types of microbes. Okay, so just to compare and contrast again, the asshats with the stealth pathogens, okay? Pathogen survival depends on spread to new hosts. In this case, the stealth pathogens, that's not their goal. They don't want to reproduce rapidly. They don't need to go to new hosts. They just want to get inside you and stay there forever, happily ever after. But because many of them are spillover from animal reservoirs, when they get to us, our immune system sees them as a new thing, However, they can turn off the immune system because they've already done this with their previous hosts. We see them, we don't know quite what to do with them, but we're not going to be able to fully attack them and get rid of them. And so it's this uneasy period that will go on for a couple thousand years probably before those things that are now causing chronic illness in us will become our commensals like they are in animals. But this process has been repeated. It's repeated a lot, right? So antibiotics are effective. Antibiotics are not effective. Antibiotics work best against rapidly reproducing microorganisms that are rapidly taking things in from their environment. That's how the antibiotics get through to them. These guys don't reproduce quickly. They reproduce slowly. Sometimes they don't reproduce at all. So they are tolerant to antibiotics. So that means our frontline drug, and for the most part for infectious diseases, the only drug, is not going to be effective against many stealth pathogens. Frontal pathogens engage the innate and adaptive immune responses, right? So as a result of that, you get outward symptoms and signs. The hallmark of being able to diagnose a disease is you need an objective sign to be able to do it. Frontal pathogens do that. Stealth pathogens do not. So they not only just maybe get the innate system is activated, but they turn off the adaptive system, immune response. And the symptoms are due to inflammation, which is a symptom, not a sign, right? So a fever is an indication that something's going on. If my shoulder hurts and I tell, my physician my shoulder hurts, he'll say, well, how much does it hurt? And I'll say, it hurts a lot. But what if I'm a whiny person? What if every little thing is dramatic? Or what if I am a stoic person and it's like my arm could be falling off and I'll be like, oh no, it's fine. So that's one of the reasons why, there's that delineation between symptoms and signs. But the point is, stealth pathogens don't cause overt signs. They cause generalized symptoms. So they can be, there's no particular clue saying this is related to that disease. Immune system typically eliminates the pathogen for a primary pathogen, but not in the case of a stealth pathogen. These guys elicit long-term memory of the immune response. These guys don't. So whole different strategies when it comes to their survival, which of course impacts how we function as well. All right, so what are the strategies of stealth pathogens? So this is an area of research. I gave you an article to read, which I hope you'll read, about stealth pathogenicity and some of the tools that these things use. But generally speaking, what we know pretty well at this point is that their goal is to establish permanent infection. They want to get in, they want to stay put, they just want to reproduce, they want to do it their way. If we respond to it, or, the goal isn't to expel them, it's just to, they're thinking, and well, I'm trying to stay quiet here. It's your immune system's problem with me that's causing it. So how do they do that? They have variable periods of growth. Sometimes they're reproducing, sometimes they're not. Sometimes they actually morph into different shapes. Sometimes they hide out in structures inside of cells and inside of tissues. Then they turn genes on, turn genes off, and manipulate the host immune response. So the ones that we are pretty well versed with, that we know stealth pathogens do, they grow in biofilms. And I got to tell you, many frontal pathogens can grow in biofilms too. In fact, most bacteria in the real world grow in biofilms. They do not just, hang out as single cells and colonies. They grow in communities, and they often form structures or multicellular based structures. So here's another one. Like I said, they can go dormant and they form persisters. There's a genetic response where particular genes are triggered called the stringent response, where it's like stringent means, the conditions aren't great, right? So somebody turned the temperature up, so I might die. I better shut back down and go into hiding. Or stress or other starvation will trigger the stringent response. And these types of pathogens will just go into a protective dormant state where they are difficult to detect and nearly impossible to treat with any kind of antibiotic because they're just not metabolizing, so they're not bringing the antibiotic to a place where they can be killed by it. Another trick is to go find a nice little hideout where the immune system, you know, doesn't necessarily have a big presence, right? Protected tissue sites. So what do I mean? The brain is a really perfect example of that. Connective tissue, which is not very vascular. If you can get out and get yourself into a joint, or, you know, the meninges, of the nervous system, or the cardiac membrane, that of the cardiovascular system, these are great places to go and hang out and start to reproduce. So wherever there's a lot of connective tissue, because connective tissue, remember, is not highly vascular, that's a great place to go find yourself a little niche and to hang out in. And then intracellular growth. So they invade and reproduce, live inside of other cells, our cells, which means the immune system comes along and looks at our cells and says, all right, you know, nothing to see here, move on. So it's a great hiding place if you can actually do it. Are there others on this list? Yes. And, you know, just keep checking back because there'll be more and more to add to this list, the more we know. Okay, so let's look at biofilms first. Biofilms, you know, bacteria that live in biofilms, some of them produce the biofilms, right? So those types of cells, and a really classic example of that is Pseudomonas aeruginosa, in fact, and other Pseudomonases, which you met in the lab. They have two very different phases of life. They have a planktonic phase where they're motile, they have flagella, they're swimming around. And then when they find a surface that they can settle down on, they settle down, attach, turn off the genes for motility. So they get rid of their flagella. They just kind of hang out there, and they turn on genes that allow them to produce their extracellular matrix, like capsules, right? And then also slime layers. So they're producing these gooey polysaccharides, and sometimes they produce them quite a bit. And then as they do that, they, you know, so they reproduce, they form microcolonies, they start to secrete this slimy stuff that they then build into city-like structures, right? So they build skyscrapers, they build low buildings, they have highways, they have channels, and that is what's referred to as a biofilm. So think of it as Manhattan, all right? So the perfect example of a bacterial biofilm is Manhattan. So imagine all those different structures, all the different roads, all the different people. Those are the bacteria going about their lives, going to find food, processing the food, all the rest of that. That's what goes on in a biofilm. And then at some point, the biofilm senses that it's time to go take over someplace else. So the bacteria will emerge from there, put their flagella back up, and start swimming around until they find new surfaces to colonize. And the thing is, in a biofilm, bacteria in biofilms are next to impossible to eradicate with antibiotics, or at least not to the point where you wouldn't also kill yourself by taking that high of a dose of antibiotic or an antibiotic for that long. So many surfaces in our body are inhabited by biofilms, and I would say probably most are inhabited by biofilms. So, you know, when you wake up in the morning and you go brush your teeth, and they all feel so nice and clean and fresh, and you rub your tongue over them, and they're, you know, you can kind of feel, and then later in the day you do that again, it's like, oh, things feel slimy. Because there are biofilm bacteria in your mouth that are producing these mucoid polysaccharides. You kill them, you know, you get rid of them by brushing and toothpaste and mouthwash maybe, but they're not gone. So you wash them away, and what they do is rebuild the biofilm in a few hours. So, but here's the thing, if you feed them sugar, they like that better, so they'll grow faster. So avoid sugar, which will help your oral cavity. Yeah, like that's gonna happen, right? Okay, so what are the benefits for bacteria that live in biofilms? Okay, so it's like, it's living in a city where everything is right there for you, right? So you have, you know, lots of diverse areas to live. You have resources. You have the ability to, you know, work with others. So in other words, here I am, I'm a bacteria, I'm going to ferment this stuff, I'm going to produce this chemical. The next guy over here is like, oh, I'll take that raw material and I'll use it, I'll ferment it, and then I'll hand it on down to the next one. And then, you know, just generally, you really just kind of think about what I said about New York City, right? Manhattan would be a perfect example of a biofilm and all the benefits and, by the way, problems that go along with that, because there is competition among the different species there. And, you know, this is perfect because they build this city. It starts off as a slime city, but then it becomes harder and harder and harder, right? So that's why when you go to the dentist and you have those biofilms, they become calcified and they have to scrape them off with that tool that makes that awful noise while they're doing it. Okay, so great life, great way to actually survive. So here's in biofilms, the persister cells were first noted in biofilms. Okay, so I just want you to kind of, this is the bacterial growth curve. Vegetative cells, meaning the ones that are actively metabolizing and reproducing, are in blue. And then notice that as we move from lag phase, where it's early in the population growth, to the log phase, as we move into mid-log to late log, notice that these persister cells start showing up. And the further we go into the stationary phase and then into the decline phase, more of those persister cells show up. So what are the persister cells? Something called persister, what do you think it does? They persist. So point being, you know, if you have a population of bacteria with 1 or 2 persisters in it, if you kill off the vegetative cells, and remember that persister cells are tolerant to antibiotics because they're non-growing, that population will increase. And the thing about persisters is that they are not dead. They are living cells. They're just not doing anything. They're not eating, breathing, reproducing. They're just kind of there, but they can reactivate. They can come back to life and restore a population. So leave behind 1 persister cell, it will rebuild the entire population, which you can see in these various graphics here, okay? This is the stringent response I was talking to. So this is the alarmone that bacteria will produce in response to stress. So what is stressful to bacteria? If you starve them, you heat them up, or you do treat them with chemicals, antibiotics, that kind of thing. That, you know, it kills some of them, but it puts the others into a metabolically dormant state, okay? So they are antibiotic tolerant. And it turns out that probably all bacteria to some extent have this ability. Some are way better at it than others, and the stealth pathogens are masters at this process. So introduction to this guy, this is Dr. Garth Ehrlich, who worked with the man that I will call the father of biofilm science. That was William Costerton. He was at Montana State University. Garth Ehrlich, you know, became famous because he was the guy who said, with regard to otitis media in children, was that these are not, so, you know, kids get recurrent ear infections. And ultimately, if that happens enough, what they do is they put tubes into the ear to allow for proper drainage. That's necessary because of the continuing infection cause inflammation, causes the eustachian tubes to completely block up. All that fluid builds back up. It causes pain. So what do you treat a kid with an ear infection with? Antibiotics. And some parents will tell you that as soon as the kid goes off the antibiotics, the infection comes back. And up until, you know, I would say the mid 2000s, there's new infections. This kid is getting these new infections every single time. It isn't a recurrence of a previous infection, or it isn't them surviving the antibiotics. That can't possibly happen. So he's the one who took on the medical community and said, here's what's happening. The bacteria in the middle ear are growing in biofilms. The antibiotics are killing some of them, but not all of them. So the kid goes off the antibiotics and the persister cells reactivate. They form a whole new colony. The whole process starts all over again. And so the tubes of the ears are basically a way to relieve the pressure and the inflammation and decrease the number of bacterial cells, which is somewhat effective. So this little 6-minute montage is something that you should watch. It's all about bacteria and biofilms and what makes them different. It has actually several different doctors in it, but it, you know, I just wanted to point out this guy because he is, again, the guy who said infectious diseases can be caused by stealth bacteria in biofilms, and antibiotics are not the answer to that problem. Do we have an answer to that problem? No, but we know that antibiotics are not the answer. So maybe we should start looking for answers. So I'll let you watch that. I won't show it to you right now. And then this gentleman is Dr. Ed Breichwert. This is from the article that I had you read from lab. He is the world's expert on Bartonella and the stealth pathogenesis tactics of Bartonella. He's actually a veterinarian. He's at North Carolina State University in their College of Veterinary Medicine, but he also has an appointment in the medical school because this is, again, a zoonotic, an animal-associated disease. That's where he first came in contact with it. And it is becoming apparent that this was a disease of veterinarians. Bartonellosis affects veterinarians who are obviously at high risk of exposure to animals that would have a Bartonella infection. So that caused a crossover for him into the medical field, and he's the go-to guy for all things Bartonella. Unfortunately, the medical community hasn't caught on that this is a remarkable pathogen that infects people in a stealthy fashion. So people have low subclinical or even asymptomatic infections with this thing that can cause problems later on down the line as the immune system becomes less effective with age or with immunosuppressive drugs or a result of other infections. And I will tell you that Bartonella is often a co-infection associated with Lyme disease, with Borrelia burgdorferi. So we're unraveling this a little bit at a time, but again, this is from that article that I had you read for the lab on stealth pathogenesis. So please check it out and realize that these are the people doing the research right now that are blazing the trail. Okay, so why is Bartonella so fantastically pathogenic? And the answer is because I would say this guy right here is probably the GOAT of stealth pathogens, GOAT being greatest of all time. Forgive me, Tom Brady, for saying that, okay? So they infect the endothelium, they infect blood cells, and what Dr. Breichwert and his team are showing is that once infected, they are everywhere. So the blood carries them to all kinds of tissues and organs, and they are able to survive successfully in all of them. This is a practically impossible infectious disease to treat with antibiotics because they go to so many different locations and because they are tolerant of most antibiotics. So just a couple of the conditions that are associated with Bartonellosis. This is bacillary angiomatosis. So these are angiomas that are due to vasoproliferation as a result of this infection of the primary niche, which is the endothelium of blood vessels. Right, so how do they get into the blood? So this is a blood-sucking arthropod. So who are these guys? They're fleas. They are lice. And we also, now there's significant evidence that this is a tick-borne. They can be transmitted by tick bite also. So they get into the endothelium, they get into the blood. From the blood, they go everywhere. This was first noticed in HIV patients back in the 80s, that a high proportion of people that had the immunosuppressive condition of HIV/AIDS showed up with these lesions, bacillary angiomatosis, and they were tested and found to be Bartonella that were causing it. This is in somebody with a fairly reasonably healthy immune system. These are striae. A rash that looks like a claw, something gave you a claw mark, again, due to vasoproliferation and inflammation of the lining of the blood vessels, right? This is why Bartonellosis, an older word for it, and the best known of the diseases is called cat scratch disease, for two reasons. Number one, because it was presumed to be transmitted by cat scratch, which it can be, but also because the appearance of the striae looks like somebody took the time to, you know, give you a good scratch. So a very remarkable microorganism. Want to point out that these guys, looking at this hemangiosarcoma, this is a type of cancer that is driven by Borrella. Okay, and again, notice the authors on this paper, Dr. Breitschwert and his team, are on here on this paper that was published most recently, showing how prevalent this disease is. Again, he's a veterinarian, so they're showing in animals, but what we need to do is the same sort of investigation with humans, because any humans that are associated with animals are at risk of this disease, and it can cause significant problems, including cancers. Another one I want to mention is Borrelia burgdorferi, which is the cause of Lyme disease, and other Borrelia species which cause relapsing fever. That's the name of the disease. So even though the symptoms are very, very similar, because there are different bacterial species involved and because of all the rest of the stuff that goes on with this bacteria, Borrelia species in general are kind of broken into those two groups, the Lyme disease group and the relapsing fever group. But the point is, they all can do this stuff, okay? So this is Dr. Monica Ember. She's at Tulane University at the National Primate Center and is doing research with Borrelia burgdorferi with non-human primates, and has been able to show all of the different mechanisms, probably not all of them, I bet there's more, mechanisms that Borrelia burgdorferi in particular, a spirochete bacteria, cause of Lyme disease, they're able to actively suppress the immune response. Now think about that. HIV suppresses the immune response. So in other words, here's the knob on the immune response, turn it off. Or evade the immune response. And these are the known strategies. So these are ones that we have been able to show. And from her talk, from this talk here, this sentence says it all. The spirochetes are suitably adapted to avoid or retaliate against every tier of the host response. Meaning as soon as that bacteria gets into you, they are off, they are evading the immune system. They also travel to protected niches. They like the heart, they like the central nervous system, right? The membranes lining that. They like the joints, rich in connective tissue. They bind there, they build biofilms, they drive inflammation. The immune system comes and tries to destroy them, can't do it, but the immune system keeps calling up more and more reserves, leading to more and more inflammation, causing damage, and that damage can actually become systemic. Okay, so that is as a result of persistent infection by Borrelia burgdorferi. All right, so the constant presence of low numbers of these things is enough to drive long-term disease. So looking at, it's not all about bacteria, even though you know how I feel about that. There are other pathogens. In lab, we looked at a few eukaryotic parasites. If you remember, we looked at Babesia, we looked at Toxoplasma, Plasmodium, right? Those are all protozoa in that apicomplexans group. There are other protozoa that can do that, but these, one thing I hope you remember from lab, Plasmodium, Toxoplasma, Babesia, live in blood, right? Where you have this huge proportion of white blood cells that are there patrolling, looking for these things. They are growing and reproducing in blood, okay? So that's how remarkable these things are. And they're transmitted by, again, blood-sucking insects and arachnids like ticks. So there are several fungi that have stealth capabilities too, right? So coccidioides, pneumocystis carinii, which, by the way, was identified as a co-infection of HIV, meaning, HIV causes immunosuppression, and then these other infections step up and are able to reproduce to large numbers and cause actual disease, okay? So on the side of the parasitic worms, we actually looked at Dirofilaria in lab. That's what the red signifies, by the way. And by the way, this right here is, these are microfilaria in an eye. Oh my God. That's all I'll say. Okay, so we have bacterial stealth pathogens. We have eukaryotic stealth pathogens, protozoa, yeast, molds, worms, which are animals, by the way. Okay, so worms are actually they're animals. They're part of the animal kingdom, which is why it's difficult to develop therapeutics, drugs for them, because drugs that kill them also have toxic side effects on us, because we're all, we're a little bit closer related to them than we are to these other things. And then there's viruses. So we have a very conflicted relationship with viruses. So something that I want everybody to understand is that our genome is, human genome, not that of all organisms. Let's just look at the human genome. So you look inside our cells, you take all the DNA out, you look at all the genes. So we have, 30,000 genes. All the rest of the gene functions that occur in cells are genes coming from elsewhere. That's a lot of it, the microbiome is doing that, but there's a lot of DNA in our genomes that derives from viral infections, past, ancient, recent viral infections. Okay, so looking at the human genome, so in a bacterial genome, we did this with E. coli, right? So E. coli is mostly genes that produce proteins and with very little stuff in between. The human genome is, there's the genes for proteins right there. 1.5% of our genome are genes that lead to protein. That means that 98.5% of our genome is not doing that. And a good proportion of it, so some of it is introns, those intervening sequences in genes. There's these random sequences. Heterochromatin is what helps the DNA fold up. But then you start looking at this stuff. Retrotransposons, transposons, repeats, duplications, signs and lines. These are all virally derived sequences. So how does that happen? Because viruses have genomes, nucleic acids get copied into new nucleic acids, and then they insert themselves into the genomes, and then the genomes are replicated and replicated and replicated as the thing continues. If the germ cells this occurs in germ cells, then that gets propagated to the next generation, to the next generation, and the next generation. So that means in our DNA, we have this vast quantity of viral sequences, number one. Also, we have viral infections that are persistent. Like, for example, all of us, I will bet you, are infected with one or more herpes viruses, and they're there. They have remained there. They will be there. They will be there when you die. Active viral infections, new ones that we don't know what's going on. Here's interesting stuff. Bacteriophage. Remember, these are viruses that infect bacteria. So our microbiome, or microbiota, have viruses that actually contribute to our virome, right? All the viruses that make up our virome are just like the bacteria that make up our microbiome. So the human endogenous retroviruses, that's these, right? So we have endogenous, meaning within us, retroviruses. Remember, HIV was a novel retrovirus. That was a new one that came, but we already have these kinds of retroviruses. All animals do. And many suspect that HIV came, and actually I think that's pretty well proven, was a simian type of virus that was transferred over to humans as a result of exposure to those animals. So, we are persistently infected with viruses, and viruses, have their own pluses and minuses as far as what they're doing for our genomes. Okay, so, some viruses, and we'll just, since we're living in the age of COVID would be one of them, provoke a profound immune response, especially the first time our immune system sees something. The reaction is, we don't know what's going on, so pull out all the stops. And sometimes that can be fatal, right? So that cytokine storms, which is what that means is your body responds with this huge innate response, produces all these cytokines. Cytokines are messengers to the immune system, so it's trying to alert everything in sight. But the problem is it also causes damage to tissues. That damage can lead to secondary bacterial infections and other complications, like systemic complications later on down the line. So those kinds of viral infections, are the ones that we all worry about. They're the ones that make us sick very rapidly. They can cause all these different types of pathophysiological effects, leading to, again, secondary bacterial infections. Fun fact about the 1918 influenza pandemic, the last big one, well, actually it wasn't, but the one that everybody keeps equating this pandemic to. In that pandemic, the major cause of death was secondary pneumonia by bacterial infections. And remember, in 1918, there were no antibiotics. So there was the influenza that caused the damage, but deaths were due to bacterial pneumonia. That was the primary cause of death for the vast majority of the millions of people that died from that particular strain of influenza, okay? So these are definitely, you know, bad guys. But what happens is after that first initial introduction to those types of viruses, you know, the survivors of us are actually prepared for the next time around, and the next time around, the next time around. So you'll notice there are no viruses that stay super pathogenic for a really long period of time. Even Ebola, when we had that incident back there in the early 2000s with Ebola coming to this country, you know, the rate, nobody in this country, other than that very first person that came here, nobody died because we've gotten to the point where we can prevent mortality. The mortality rate was almost 100% when Ebola first emerged, but now it has backed up. So the mortality rate is much lower. And in this country, it's even lower because we have therapeutics. We have approaches to therapies to deal with those kinds of outbreaks. Okay, so bad guys. Realize that many viruses establish latency, and then they can reactivate later. And case in point are the human herpes viruses. The one that I'm looking at here is human herpes virus 3. You may know it best as the varicella zoster virus, chickenpox, shingles. Varicella is chickenpox, zoster is shingles, the VZV virus. So what happens with this human herpes virus 3? At a young age, you get infected with it through usually a respiratory route. And it ends up localizing in skin, and what you end up with is breaking out in pox, right? Chicken pox. I don't know why they're called chicken pox, but they're spots, you know, gooey looking spots all over your body. Those spots rupture, and those have infectious viruses in them, which is how it can get spread. And then, you know, basically the pox heal up, and then you have long-term, lifelong immunity to future infections with varicella, new infections. However, what these viruses actually do, so most kids, by the way, don't end up with severe disease. They get chickenpox. They, you know, it's not very comfortable. It's not a great disease to have, but when they recover from it, they emerge with lifelong immunity. So, but what happens is, for probably most everybody who's infected with this virus, the virus moves from the skin into the nervous system, and once in the nerve endings, it actually travels up to the nerve cell bodies, right? So actually the dorsal root ganglion is where this virus creates what's referred to as an episome. So it just creates a little space there, and it sits there for a very long time, 20, 30, 40, 50 years, sometimes forever, without there ever being a secondary complication from it. But in some cases, if there is a failure of the immune system, so if you become immune suppressed, or as we get older, that sort of happens anyway, the infection in the dorsal root can reactivate, meaning it goes back down the axon, out to the axonal terminals, crosses over, gets back out to the skin, and it begins the process all over again. But realize that this widespread chickenpox, what the viruses do is localize usually to one or a couple nerves. So, you know, if it's a cranial nerve, then you could have, you know, the skin lesions would appear wherever those cranial nerves emerge. If it's a spinal nerve, then they will show up whichever spinal nerve is harboring the latent viruses, right? So this is, you know, this is what happens. You get shingles as you get older, which is called zoster, okay? And then some of these cases are very, very severe, right? If more than one nerve was infected or more than one nerve is reactivated, you can have severe nerve pain as a result of that, but most people don't. So here's an interesting thing. So there is a chicken pox vaccine. There's a shingles vaccine, okay? So the point being, instead of kids getting chicken pox and developing this long-term, lifelong protective immunity, the chicken pox vaccine does not provide that. So what it provides is short-term immunity, enough so that if you are infected, there's still a chance that this can happen, that the virus, so in other words, you would not experience severe disease as a kid, but you can still end up with latent infection in your dorsal root ganglion, which will reactivate within 5, 6, 7, 8, 9 years. So what's happening is an increase in the number of kids that are getting shingles that have been vaccinated. So they're protecting against the first stage, the chickenpox stage, but promoting shingles through that vaccine. So that's kind of an interesting conundrum when, you know, it's like, well, should I let my kid get chickenpox or do I want him to get the vaccine? And that decision is being taken out of our hands. So it's sort of like, do I want my kid to have lifelong forever immunity and hopefully never end up with shingles, or do I want them to get shingles? Nobody wants shingles, trust me. Had it, hurts like hell. You don't want shingles. So just, you know, these are things that are not thought about deeply because everybody thinks, oh, let's prevent all diseases. Well, okay, but we're causing a new set of problems by doing that. And that is the part that doesn't always get inserted into the calculations when the time comes. Okay, so looking also at Epstein-Barr virus, HHV4, and also HHV5, which is cytomegalovirus. The acute condition is called mononucleosis. It is a disease of profound fatigue, among other symptoms. And again, this is a virus that can establish latency. So, you know, the original infection is usually a respiratory route, starts the infection. But notice that when they get trafficked to the lymph nodes to start the adaptive immune response, what these viruses do is to establish a latent infection in B cells. So B cells are the cell type that differentiate into antibody producing cells. So they can become persistently infected with HHV4 and HHV5 and probably HHV6, okay? So these are the memory B cells that are infected with a virus. So then when those cells go into blast stage, that can actually prime reactivation of the latent infection, leading to another round of infection, which allows them to spread. But the point being, they establish a long-term, perhaps lifelong infection within the memory B cells. Okay, the memory B cells are stored throughout your body, all right? So the resting B cells wait in the lymph nodes, and then they can release infective viruses. So in other words, shedding, it allows people to, you know, asymptomatic people to pass the infection on to other people. So why do we care about that? For a lot of reasons, all right? It's a virus. There are no treatments, really very good treatments for viruses. For herpes viruses, there are some, but they're used for the viruses that, like for example, they're used for the sexually transmitted herpes viruses. We have viruses because they do the same thing with different cell types. So once you're infected with herpes, the bad one, herpes, which is 1 or 2 simplex viruses, you are forever infected with that, and you can forever spread that to your host. But now there's an antiviral drug that prevents viruses from replicating. So in other words, it prevents this from happening. So you won't pass it to your sexual partners. You wouldn't have to be concerned about that as long as you take that drug, because you cannot ever get rid of these viruses. They will always be there. So I added this because we didn't really talk as much as I would have liked to about epigenetics. Epi is above, right? So the genetics is what's in the DNA, epigenetics is what's controlling gene expression. There are lots of levels of epigenetic control. Just realize two of them that we know the most about is methylation. So we can shut off genes by sticking a methyl group where the translation starts. And then also modifying histones, the stuff that wind up the DNA. So DNA packaging is affected and also methylation is affected. So we can silence genes. So, you know, you can silence them, but the point is they can also cause reactivation. So what we're finding is that these, you know, these latent herpes viruses, okay, in particular, you know, Epstein-Barr virus is the one that everybody tends to look at. The primary is asymptomatic or has a self-limiting disease, but then just realize that both of these things, they elicit a strong immune response, but when the viral load increases, you could end up with autoimmune type diseases because the immune response that gets triggered, the immune system responds to the cells that are affected, and that can lead to what they call molecular mimicry, meaning that the response to the pathogen is cross-reactive with something within us. So we have antibodies attacking our cells, and that is the pathology associated with multiple sclerosis, other autoimmune diseases like lupus and RA. So in other words, these can be, so is the virus causing the disease? And the answer is, well, okay, it triggered it, but then there's more to it than just that. So it isn't a cause and effect. There's a cause, and then the effect is greater than the whole is greater than the pieces, for sure. All right, so just, you know, a little bit more on that. So it turns out so it's like, I want to hate Epstein-Barr virus, right? I want to hate those herpes viruses because they're doing all this to us. But it's not that simple. So they cause all those issues, but on the other hand, notice that, you know, we have come to an equilibrium, if you will. Humans and these viruses and animals, which have their own sets of herpes viruses, have you know, this balance. And when it becomes unbalanced or altered, autoimmune diseases can be the result of that, right? So notice, you know, all this stuff, this is a pretty complicated little picture here, but notice, so here's what the herpes viruses does. You end up with cross protection against other pathogens. Being infected with herpes viruses offers you protection from other things. It protects you against lymphoma. It protects you against autoimmune diseases. But when it gets out of balance, like when a high viral load occurs, like reactivation of those latent infections, then you have the bad effects. So we have a positive relationship, but we also have a negative relationship. And that is one of those things that is not taken into consideration all that much. When we look at autoimmune diseases, how do we treat them? We suppress the immune system further. So isn't that going to throw the viral, you know, this relationship we have with this microbe and probably our microbiome, isn't that going to throw that balance even further out of whack, leading to continued disease? So we're treating the symptoms, but we're furthering the disease. And that is unfortunately the model of treatment for infection, for autoimmune diseases, for immune system related diseases, chronic illnesses in the country. So you treat the chronic illness, you treat the symptoms, you want to lower the inflammation, you want this, you want that, but at the same time, you are promoting a continuation of the chronic illness. So, and if that wasn't bad enough, you know, I just want to point out that, you know, there are cancers that are directly associated with Epstein-Barr virus. So, you know, of the 96% of the human population that is infected with Epstein-Barr virus, HHV4, how many go on to develop Hodgkin's lymphoma? It's a very tiny fraction. So there's got to be secondary or tertiary factors, some of which I just talked about. But another one that we aren't talking about is an infection of the B cells. So the memory B cells are already infected with Epstein-Barr. When you add in another pathogen that infects the same cell type, like Mycoplasma fermentans infects the same type of cell, guess what happens? You trigger immortalization, all right? So you trigger changes in the genetic makeup of those cells. They become immortal, meaning they don't go through apoptosis, they start to proliferate, they get out of control. That's cancer. So immortalization of a cell line is phase 1 of any kind of cancer. So the lymphomas that are associated with Epstein-Barr infection are probably not caused directly by the Epstein-Barr infection, right? The latent infection is caused by a co-infection by something else in the same cell that completely disrupts the genetic controls over cell division within that cell, leading to out-of-cell control growth. Okay, and you can see the same relationship here between Helicobacter pylori and Epstein-Barr virus leading to gastric cancer. So before we started taking antibiotics widely, probably every human was infected with Helicobacter pylori. And then in the 1980s, they figured out, these ulcers are caused by this particular bacteria. So they started treating ulcers with antibiotics and other things. And as a result, the load of H. pylori has gone way down. But the point, and that's a whole nother story, because H. pylori probably had a role in our stomachs as well, but now we've sort of eradicated all the H. pyloris. But the point being, not everybody that had H. pylori developed gastric cancer. So here's a model indicating that the H. pylori infection along with Epstein-Barr and probably other factors too, can lead to different types of gastric cancers. So it's not just one thing causes one disease. It's multifactorial. And our treatment approaches to all these diseases are not multifactorial. They're usually, what antibiotic can I use to kill off H. pylori without considering, again, what comes downstream from that. Okay, so I think, at this stage of the game, actually, this is just the abstract, part of the abstract from that. Notice the rate goes from Epstein-Barr virus positive B cells co-infected with mycoplasma, 74% became mortal, meaning they were headed for cancer, versus 70% of only the Epstein-Barr infected cells. So it's the co-infection that actually is what triggers the cancer, not just the one thing, but multiple factors. And then I just want to point out too that we now know, based on research, that Borrelia burgdorferi does this too. So Borrelia burgdorferi is taken by the phagocytes to the lymph nodes. In the lymph nodes, they completely disrupt the germinal centers where all of this training of T cells by B cell and T cell training occurs. They completely screw that up. And so the question is, in Epstein-Barr positive environment, since they establish latency in B cells, could it be that when Borrelia shows in there and like plows through the lymph node itself and tears it all apart, could that be outing, if you will, right? Pushing it out from latent into, an active state again, an active infection, and causing high viral loads, which you already saw that high viral loads are what are associated with all these issues. So can Lyme disease do that? And the answer is yes, Lyme disease can do that. So for what that is worth, check for ticks and kill them if you find them. Okay, so speaking more about Lyme disease, in addition to for Borrelia burgdorferi, because they already mess up the immune response on their own, what has, again, increasingly understood is that Lyme disease is not just a single infective disease. It's not just one thing, that there are not just co-infections with viruses, but also different types of bacteria, right? So notice this is just an indication. This is a serological investigation, meaning they're just looking for antibodies. But notice in a population of patients that have tick-borne diseases, number one tick-borne disease is Borrelia burgdorferi, but notice there's evidence of infection with all of these other microorganisms, particularly in the most severe cases. So think about that, all right? So that's exactly the situation that happens with HIV. So HIV goes in and infects the helper T cells, causes them to get all screwed up. Borrella burgdorferi goes in and doesn't, it stops the helper T cells from proliferating. So the helper T cells will start off and then they just go, which means the B cells have no instructions, so they don't know what antibodies to make or what to attack or anything else. So it's not the exact same mechanism as HIV, but it's the same plan. It's the same strategy as HIV. So they're not infecting and invading T cells. They're just stopping that whole line of communication between T cells and B cells. So the immune response is not long-lived, it's short-lived, it's an immunosuppressive state. And if the infection is allowed to persist, that immunosuppression goes on for a very long time, followed by secondary co-infections. And that's exactly what AIDS is. Okay, that's how you diagnose it. A white cell count below a certain stage, HIV positive, and then a list of co-infections, bacteria and many yeasts and other co-infections that will lead to a diagnosis of AIDS, right? HIV positive is not AIDS. AIDS is a disease condition characterized by being HIV positive and then having all these other co-infections to go along with it. So guess what? That's what Lyme disease is too. Okay, so at this stage, I hope you appreciate that it's very, very complicated. Microbiology used to be the study of microbes. It used to be studying bacteria grown in culture, and it has morphed into the study of how microorganisms interact with, all of their various hosts. I'm just picking on humans because I happen to be one, but they play a critically important role in keeping us healthy. So we need to broaden the scope of microbiology, which microbiologists are, by the way. Medicine, not so much. Microbiologists are, to encompass all of that, the role, the total role, the goods, the bads, the uglies of microorganisms in terms of human health, and study it in the context of health and disease, and figure out ways to mitigate this epidemic of chronic diseases that humans in countries like this one are actually undergoing. So that concludes my talk on stealth pathogenesis. Hope you had fun, and I'll see you next time.